I presented a study we did aiming to try to identify patients that are prone to develop brain metastases. So this is lung cancer patients that have early stage cancer with no metastasis in most cases. They undergo surgery. So the tumor is removed and they are healthy in principle, but they're being followed up. And in many cases we know these patients with lung cancer history develop recurrence of disease. And one of the worst outcomes is development of brain metastasis, so recurrence of the disease in the brain. So if we could know in advance who are these patients, we would follow them more closely. And if we would understand further the mechanisms of the development of these metastases, potentially, theoretically, we might be able to find a treatment to prevent that. So that's the far goal. So what we did is we collected a set of samples that were taken at surgery when there's just a small tumor that's resected. And we compared these tumors between a subset of those that we had a good follow-up and no metastasis developed, and a subset of patients that developed brain metastasis later on. So this was retrospective, so we had this data. And from this comparison, we could identify a set of genes that were highly correlated with the risk of developing brain metastasis. And this was significantly more predictive of brain metastasis than just looking at the clinical data. And then we went on to try to understand the mechanisms that are involved, and we found very interesting sign of the respiratory chain, oxidative phosphorylation type of metabolism that was very high in those cases that eventually developed brain metastasis. So this is a very interesting direction. There are now compounds in development of inhibitors of the respiratory chain. The interesting point that I should stress is that the signature we found was specific for brain metastasis. It was not up in patients that developed metastasis in other sites. So this suggests that it's really a mechanism that's correlated with the process of development of the metastasis in the brain, of homing to the brain, settling there, and growing there. Well, this is not yet there, but in the future, if we validate that, if we create a tool that can be used clinically, we could be able to know which patients should be followed up closely with brain MRIs and so on. And then in the event that there is a recurrence in the brain, we can treat it early. And again, further away in the future, if we prove that the mechanism is what we suspect, and if this is related to oxidative phosphorylation, we might envision a clinical study where we inhibit this and see if this prevents metastasis in these patients.